those of you who this is your manyth time at an energy talk. My name is Valerie, and I am the Outreach and Engagement Lead for Future Energy Systems. And before I start, I'd like to acknowledge that the University of Alberta, where I am from and where Future Energy Systems is from, is located on Treaty 6 territory and the North Saskatchewan River territory of the Métis Nation of Alberta. And we want to acknowledge that this is a gathering place for many diverse Indigenous folks, including the Cree, the Blackfoot, the Métis, the Nakota Sioux, the Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant community. And many of our researchers have worked outside of this treaty area. We want to acknowledge all of the communities that have been important partners in this research. So Future Energy Systems, for anyone who this is your first time, is a research program that was launched in 2016 at the University of Alberta with $75 million from the federal government from the Canada First Research Excellence Fund to help Canada transition to a low net carbon energy economy. And we focus on multidisciplinary research, everything from how do we develop new energy technologies, how do we integrate them into the current system, how do we deal with issues for our society, our economy, and our environment, which is what we're going to focus a little bit more on today, but also how do we develop solutions for our current challenges. We have over 100 projects, over 150 researchers, and over 900 graduate students, postdocs, and other highly qualified personnel. You will notice that Carlo is in the back with the camera. We are just filming Haley and the talk. If you do ask a question later, your voice will be heard. So if you don't want that, go talk to Carlo at the end, but we won't be filming you. It is just so that for anyone who can't be here tonight, they have a chance to watch the talk. So I am going to pass it over to Haley. Dr. Haley Hunter is a recent Ph.D. graduate from the SERPA lab at the University of Alberta, where she studied smart polymers and their use in developing color-changing sensors. While pursuing her studies, Haley focused on being involved in the community and engaging people with science, which is where Haley and I met. And she is now the chemistry lab instructor at MacEwan University. Go you. So I'm going to pass it over to Haley. If you have a question during the talk, feel free to raise your hand, ask your questions. There will be lots of time at the end for discussion, for talking. But if you got one, you're like, I need to know this now, pop that hand up and we will absolutely pause. All right, I'll pass it to you.
Valerie did a lovely land acknowledgement. So all of my research was done at the U of A on main campus, so in Treaty 6 territory. Who am I? You did a great job of that. I spent 9 years at the U of A, so I did a bachelor's degree in biochemistry. So I actually did some research on breast cancer cells and how they can either metastasize and move throughout the body and what kind of proteins allow them to do that. Switched degrees completely when I decided to do graduate school afterwards, and I did my Ph.D. in Analytical Chemistry at the U of A. So I graduated in 2024, so in September, the beginning of September, I defended my Ph.D., and then I actually did my graduation ceremony this past November. I am currently a lab instructor at MacEwan University, so I got a job in July. I started teaching in September. It's been great. I love it at MacEwan. These are just some key points from grad school and what I've done. Lots of them were actually with Valerie and doing the Future Energy Systems Outreach Program. So talking at the Tell Us World of Science and all of that stuff. Working with kids is awesome.
You can also see Haley many times on our YouTube channel
yes.
if you'd like to learn about water.
Haley does not watch those because she doesn't like to hear herself, but they are there. So what are we going to talk about today? We're going to talk about sensors. So you may think you know what a sensor is. There's broad definition and lots of different sensors that are out there. We'll then kind of transition into what a point of need sensor is. Lots of them you likely already know about. How we can all benefit from them, so why so much research is going on for point of need sensors. You may be familiar with a lot of the healthcare ones, but not so much for environmental monitoring, which is kind of where the Future Energy Systems program comes into play. The research that's been currently done, different materials that can be used for sensor development, and then a little bit about the sensors that I focused my research on. So a sensor, what is it? Anyone have an example of like a sensor that you probably use on a daily basis? There's lots.
Thermostat.
Yeah, Nailed it.
Speak out loud. This is insane. Shout him out.
In broadest terms, the dictionary tells you that a sensor is any device that can respond to a sort of stimulus and transmit that to something else. So that can be something like heat. Your smoke detectors will sense particles in the air and tell you that there's something going on in your house. Microphones, all sorts of things can be considered a sensor. We're not going to talk about the wide, giant range of sensors that exist out there. We would be here all night, but we will narrow it down to what a point of need sensor is. Sometimes they're called devices, diagnostic tools, all of those different terms, but generally speaking, we can call them point of need sensors. So point of need sensor is basically any tool or device that can measure a change in whatever the sample is that you're looking at the location of where that sample is collected. So that means we can take that sensor, kind of like this portable microphone, wherever we need to use it, versus most kind of healthcare or diagnostic tools, you would imagine being in a big lab, so you'd have to transport your sample to the lab or send it in to somebody else to do that work for you. A point of need sensor you can take with you where you need to use it. The change that you're detecting in that sample can be lots of different things. It could be a change in chemicals. It could be a physical change. So there are things like pressure sensors. So when you would touch it, it will detect that change in pressure, or biological changes. So that would be lots of things in healthcare, or those COVID rapid tests and things like that. They are arising for many different areas. So like I mentioned, we won't just touch on healthcare point of need sensors. They're common for food safety, as well as environmental techniques. And like I mentioned, they can be deployed outside of a laboratory environment, so you can take them with you. They can usually be used by an untrained user, so you can potentially order it online, use it at home following the instructions, and get your results pretty rapidly. So if you've ever had like a medical test done and you gotta wait 5 to 25 days to get your results back and then it's a little too late, that's kind of where these sorts of things come into play. Why do we need them? I've kind of talked about this a little bit already. For healthcare, it can speed up that process. So traditionally, you get sick, you go to the doctor and give them a sample, then they're gonna take that sample and send it to a lab. Could be in the same building, could be in a different building. Then one person is gonna process that sample, another person could do the data analysis and figure out what the data means when they process a sample. Then they gotta send it back to your doctor's office, and then your doctor has to look at it, and then they call you and tell you what's wrong. That can be a long process. If you have a very contagious disease, then you could have been spreading that around for 5 days before you got your results back. If you're sick with something that maybe needs medical attention right away, you don't want to wait forever. A point of need sensor, you can get your sample at home, you can add it to your test. I feel like this, laser doesn't show up on here. Sorry, guys. Yeah, it like reflects off the TV. That's okay. And you get the results right away. This is also good for doing environmental testing. When we're thinking about things like our climate and how we are trying to monitor changes in our lake water, changes in our river water, changes in air quality or soil quality, if you're trying to monitor at a big farm and your different greenhouses and you want to make sure that all of the conditions in your greenhouse are being constant, or if you want to see if something changes randomly and that's when your crop goes bad, all sorts of different things. You don't have the manpower to be out there testing something every 30 seconds or every minute or even every hour usually. If you have a sensor that can be deployed and collect data while it's sitting out there, can help you basically figure out what's going on and track those changes. So if you had a contaminated body of water that is really far from the city, you could go test your body of water with your tool while you're out there, get the results, and maybe apply something to remedy that situation while you're there. Versus traditional methods would require you to drive out to the contaminated water, take the sample, drive it back into town, take it to the lab, do your analysis, figure out what's wrong, drive back out to the water, and administer any kind of remedy situation. So they have a lot of potential to make our lives easier. They can bring down the cost of a lot of problems. Disease control is a big one, both for healthcare and the environment. Time is also a big one. We saw kind of the impacts of this with the pandemic. So when our healthcare system had to manually administer all those tests, and try and figure out who was sick and when they got sick, and the labs were delayed, and then you couldn't get your test results, or they lost your test results, and then you were home for 2 weeks and you didn't know if you needed to be home for 2 weeks. Those rapid tests that you could do at home really helped alleviate a lot of the pressures that were on our system. So that's the idea here. Location is a big one, and sometimes when we live in a city like Edmonton, we don't consider this a lot. If you live in a rural area or even in an underdeveloped part of the world where you don't have access to big hospitals and big centers, these kinds of things where you could test your well water to see if your drinking water is contaminated or not, or to administer rapid health tests. These things can be super helpful in those parts of the world. And data tracking is also another big one, especially these days. It's all about data and how we can look for patterns in the data and how we can predict changes from the data. Having these sensors that could passively acquire the data without people having to be there all the time helps for that as well. There are lots of common examples. So for healthcare, we have things like at-home pregnancy tests, those COVID rapid tests. Blood glucose monitors are one we often forget about, but if you ever have met a diabetic, they will have one of these. There's things like at-home strep tests for monitoring different cardiac markers, breathalyzers. There's lots of rapid tests for drug screening now too. But they're not just popular for health. Like I mentioned, there are soil moisture sensors. There's lots of handheld water sensors that can tell you different things about a water sample. Its pH level, the amount of dissolved solids that are in the sample, which is important for environmental tracking of water. Water hardness. If you had a pool, you may have seen this before too. You used to have to monitor the chlorine level and the pH level of your pool. So they had these kits that you would use at home. Are there any questions? Yeah.
Just sensor in my mind, it's like that sensing something and give you changing the based on the some characteristic, maybe physical, chemical, or biological, will give you a response. The sensor cannot analyze the system, cannot respond, cannot data processing or post-processing. This is a device that the sensor part of this device. Yes?
That's correct. So point of need sensor is basically a term. It's just a broad term, so there are things like point of need diagnostic tools. When we're talking about basically the data analysis and things like that, a lot of that has been done in the development of the device. So then when you have something like a COVID rapid test, the analysis of whether you are sick or not is kind of built into the system. So whether you get a positive result or a negative result. So there still is all that lab work and research that goes into developing the system, if that kind of answers your question.
Yeah. Get it. Just in the control system, we say on-off system.
Mm-hmm.
Yeah, just not the other kind of controlling the system, just give you yes or no.
Yeah, so it's just telling you what's going on. Yeah, but it's not necessarily treating a problem. It's just telling you.
And the graph, for example, contamination in this system. For example, we have a H2S in this environment. Just the sensor tells us that the level of H2S is in, for example, ppm.
Yeah.
For example, it's like this, and you cannot understand exactly what's the amount and something like that.
Yeah, there's a lot of machine learning and AI work that's going on with sensors. So there's a program at the U of A right now called From Data to Design. So I believe it's 12 different professors at the U of A from the Department of Ethics, from chemistry, from engineering, kind of all over, and they're trying to work on, okay, we have these sensors or devices like this that collect this data. We have all of this data. How can we use machine learning to kind of predict and deal with the data and then tell us kind of what to do with that? That's one of the main problems of the research that I'm going to talk about that my Ph.D. group did, is we could generate a lot of data. And we were like, what do we do with all of this data? How do we figure out the message that all of these numbers and all of these pictures and everything are telling us? So that is an area. So one of the common sensor methods for these at-home tests, so something like the COVID test, are called lateral flow assays. They're basically a way that has been developed ever since the at-home pregnancy test came out, and they can be modified by just changing the chemical groups that are on the strip. So it's like a little piece of paper, and it can be modified to detect various different targets. So it's a system that's kind of been developed. That is one of the reasons we could get those COVID rapid tests out pretty quickly, because the lateral flow assay framework already existed. So we just needed to kind of tweak it and get it to work for that different target. These are good and bad. So the way they work is you add a sample. Your sample will bind to one of the, if it is the correct target. So if we were testing for COVID, you had your saliva, you kind of spit on it. If the COVID antibody or whatever marker you're looking for is present in the sample, it would then bind to these right here and would move across the strip and will give you that red line that you would see on your test strip. This is an example of how it's done for the pregnancy test. So you would have the test strip and the control strip. If your test strip was red, that means you had the protein or antibody or whatever the target was that that test was looking for present in your sample. But the problem with these tests, if you ever did one, was they're kind of yes or no. They depend on the concentration. Sometimes can you see the line? Can you not see the line? So they are very good for some things, but they leave a lot to be desired. So that's one of the reasons there's still a lot of research going on in point of need sensors, and how can we make them better, basically. There are different materials that are then being looked into to see how we can advance this field. Colorimetric sensors are a very big area of interest because it's easy for people to interact with. A color change is something that most of us can see, or we can use our cell phones to take a picture of it and tell us what the color is. That's a big area of interest called smartphone sensors right now. So they developed the little chip and you can take a picture of it with your phone and you have their app, and then their app will tell you what was present in your urine or your spit or something like that. So there are different types of materials that can be used to develop these types of sensors. The area that my group focused on are called photonic materials. Basically, what it means is there's different alternating materials that are going to interact with light in different ways, and that will change their color. So if you have, for example, in this schematic, we have this alternating structure here. If for some reason one of these changes their size, the wavelength of light that is interacting with the material will change, and then the color of the device is going to change. So if we can harness these types of shapes and materials to allow them to interact with the targets that we may want to detect, so whether that's bacteria in water or a protein in your spit that tells you you have some sort of sickness, and allow that shape change to be a response of that target in solution, then we can get a color change. One thing that I think is really cool to talk about here is it's something that's called structural color, which we see a lot in nature. A lot of research that happens is based on nature. One of the terms is biomimicry. So nature's already made it perfect, and we just kind of try to use that in the lab and see how we can use it to our benefit. So something like a butterfly's wings, fish scales, the feathers, all of these things, if we look at them at the nanoscale, they all have different photonic structures, and that's what allows them to exhibit these colors. So something like a chameleon and how it can change colors is because it can change basically like the distance between the structures in its skin, and that changes the wavelength of light that we see. So it's not necessarily changing its pigment, but it's changing like the structure of its body that allows it to change its color that we see. So there are micro or nano structure changes that allow it to interfere with light in a different way and allow us to see that different color. We're going to switch gears to polymers. So my research basically takes these photonic structural colors, integrates polymers into them, puts them together to make a device that changes color. So we're going to talk a little bit about polymers and then kind of bring it all together. Lots of you can probably name a polymer. You're probably wearing one at least right now. A polymer is basically made up of a repeating unit. So you have one thing called a monomer. If it repeats, we get a polymer chain. So something like polyethylene, you may be familiar with, type of plastic, but you have the starting chemical here. If we react it together and put a long chain of them together, we can get something that is a polymer. This is just how we write it. But you know lots of them. So there are naturally occurring polymers and there are synthetic polymers. Rubber, we can make synthetic rubber, but it was naturally occurring when we found it. Chitosan is another naturally occurring polymer. It comes from the shells of crustaceans, so like lobster, shrimp, crabs. Cotton, silk, those are both natural occurring polymers. And your DNA in your body is a naturally occurring polymer. Proteins are also a polymer structure because anything that is built up of a repeating unit can be considered a polymer. What you probably think of when you hear polymer is plastic. So that is those synthetic polymers. We have things like these plastic clamshells that our fruit comes in. You've got those plastic water bottles that are the devil. These are, I use them in my demo with the kids. So they're called Orbeez, or those like water beads you can use to water your plants. So kids love them. They make a mess when I take them places. But they're basically like a hard little plastic that absorbs a bunch of water, and then it's kind of like a squishy gel. Styrofoam, polyester, so there are naturally occurring polymers that you likely wear on your body. There are also lots of synthetic ones. But polymers can be a huge range of things. We can look at like coatings, latex coatings, paints, lots of those things also include polymers in them. I called them fancy polymers here. So my research focused on stimuli responsive polymers. That's just a fancy way of saying that they kind of work like a sensor. So you have a polymer, it's like a long skinny piece of spaghetti. It can respond to a change in stimulus and change its shape. So stimuli responsive polymers can change shape with their magnetic field. So if they're in the presence of a magnet, that touch and pressure, I keep pressing the laser, you can't see it. Touch and pressure, I'm just wasting my battery. They can be responsive to UV or light. You can have polymers that'll change their shape when the pH of the solution changes. If the salt concentration in the solution that they're in changes, they can change their shape in the presence of different molecules or antibodies or proteins. They can also change their shape when it comes to temperature. So we're going to talk about those ones in a second. I had a couple examples here of just how these are used in research. So we have a stimuli responsive polymer that can change shape with proteins that are present. So if you have some proteins that are bound in the polymer itself and you add its target proteins into solution, they will bind to each other and it kind of makes the polymer bind to each other. So that's used a lot in sensing research. You can have polymers that will change their shape with light. So it'll just affect the bonds that are in the polymer and cause the polymer to change its shape. If you put something like a magnetic little particle into the polymer network, then you can get that polymer to respond to a magnetic field. The polymers that I used were thermoresponsive, so that means they can respond to changes in temperature. They start out as a long chain when they're at a cooler temperature. So the ones I used were anything below 30 degrees Celsius, they would be a kind of a long chain. If you raise that temperature over that 30 degrees, they would collapse on themselves and form like a little particle. And that shape change is then something that we use later on to make that color changing device. We don't need to talk about that. We can then take these polymers, so these long chains, and do a specific type of polymerization or reaction in the lab that makes them form these gels. They kind of are like really small versions of those Orbeez I had on the slide earlier, if you looked at them under a microscope. So the way it works is the polymer, while it's growing, we raise the temperature, and that means that polymer wants to collapse on itself while it's growing because it's above that temperature that it doesn't like to be at. And then it kind of grows in on itself and you end up with a little particle. We call them microgels. Depending on their size, sometimes they're called nanogels. That is usually up for debate depending on who you talk to, what that size threshold is usually. But we'll go with microgels for today. So we have these cross-linked polymer networks or microgels, and they're also going to respond to changes in temperature. So when they're at a low temperature, they're nice and swollen. They really like water, so they get nice and big. And then when you raise the temperature up above that threshold temperature, they decide they don't like water anymore and expel all the water out of the particle, and the particle will shrink. We can actually see this visually. So if you look at this solution at low temperature, it's pretty translucent. You can see through it. When we raise the temperature, the solution looks really cloudy. It's because all the particles have shrunk down, and then they're going to diffract more light, and that's why you can see them in the solution.
Okay, can I have a question?
Yep.
This is a reversible process?
Yes. Yes.
And this is destructive for the material or not?
It's not. So it's a hydrophobic collapse that the polymer undergoes. So the functional groups on the polymer just go through a hydrophobic collapse at that temperature. You can have polymers that do the opposite. So you can have polymers with this is called a lower critical solution temperature. So it goes from a one-phase system to a two-phase system. You can have the opposite with an upper critical solution temperature, where it will go from a two-phase system to a one-phase system, but it is a completely reversible process.
And the condition of the maintaining this sensor should be controlled, yeah? Because this is a gel, maybe the water comes out of the gel and the structure ruined before that you use it, yeah?
Yes, and there's different modifications that can be done to the polymer that I will talk about in a minute that can allow it to have this, basically this response to things that are not temperature as well. So if we change the functional groups or the chemicals that are involved in the polymerization in the chain, we can make the polymers respond to different things such as salt. We then, to make these color-changing devices, take these microgels, these particles, and incorporate them into one of those photonic devices that I mentioned earlier. The color is very faded on this screen here, but what we have is a thin piece of glass. We deposit a layer of gold. It's really thin, so it's about 15 nanometers of gold, so we're not talking a ton of gold. People always hear gold and they think, Haley, these sensors are going to cost $1 million. What is the purpose? We then spread a layer of those microgel particles that we just talked about making, and then put a second layer of gold. So it's like, I talk about it like a sandwich when I talk to kids. I should have brought my little demo. Kind of like peanut butter sandwich. So the gold is your bread, and then the good stuff, the peanut butter, is that microgel layer on the inside. The group that I did my research in at the U of A has been working on these types of devices for about 12 years now. So there is a lot of kind of the ground research has been done. We're making lots of improvements these days now that we can do image-based analysis. It's a big thing. But they have been done so with that temperature change that I talked about on the last slide. So when the device is heated, those particles expel the water and the whole thing shrinks. And then when we cool it down, they absorb water again and will swell back up. There's been some research that was done looking at pH changes. So you can see the maple egg there changes its color when the solution is in an acidic solution or a basic solution. There was also some research done looking at making these devices sensitive to glucose by adding different chemicals into the polymer structure that can then bind the glucose and change how much water is in the microgels. To make these polymers, the ones that I'm talking about, required three things. So we use N-isopropyl acrylamide. That is that polymer that responds to temperature. It's a very well-studied thermal-responsive polymer, especially in the healthcare field, because it changes its shape close to that body temperature range. This molecule here called BIS just helps us make these particles because it can bind two polymer chains together. It's called a cross-linker. And then we incorporate a little bit of acrylic acid. That is a pH-sensitive group. It can also be used to help bind other things like proteins into the polymer as well. So we add all of them together. It's pretty simple. We just kind of heat them up and add an initiator. So this is called ammonium persulfate. It will start that polymerization once it reaches the 70 degrees. And then we let it run for about 3 hours and filter it and get our particles out of it. We then make the devices with them. So we take clean glass slides, we deposit the bottom metal layer, kind of spread a layer of those microgels on there, let it dry, rinse all the unbound stuff away, and then deposit the top layer to get that sandwich structure. A lot of the research that I did involved this top metal layer and kind of playing around with it. We'll talk a little bit about it, but I won't bore you with all of that because there's a lot of stuff that is not very exciting.
You use the gold because it's very conductive?
We use gold. You can use other metals. So there has been research in our group looking into using things like titanium, chromium, other types of metals. The gold gives a really nice reflectivity, so we get good light coming out of the device. That's one.
The substrate is the substrate glass, chromium,
Yeah.
gold,
Yeah.
gel, gold.
Yeah.
Inside light, then outside. Yeah.
Yeah. I'll have a picture on this slide. So this is pictures of the actual devices. So they're just little squares. We can make them different shapes depending on how you pattern the gold layer onto the glass. But this is kind of the color, some of the colors we're looking at. The bright pink, this is more green than it looks on that screen, but you can get a variety. The color will change depending on how big those microgel particles are that you put in them. This is kind of what they look like on this is using an electron microscope. So these particles, the circles, are about 300 to 500 nanometers. So they're very small, packed on there. But you can see how nicely shaped they are and how nicely packed they are. We need them to be nice and packed like that to get color. Some of them, pictures I show, I don't know how many pictures I have, you may notice that the color is not perfectly homogeneous or the same throughout the whole thing. Lots of that will come from any impurities in the microgels themselves and how they've packed on the surface. So there is people in the group kind of working on that and optimizing that to make sure we can get nice, good color on the whole thing. But kind of what you talked about the light. So this is the light will enter the cavity, it'll bounce around in the cavity and then come back out. And the color that we see will depend on the wavelength of the light. And when we kind of keep everything else the same, the wavelength of light that comes out is going to be dependent on the distance between the two gold layers, which means the distance or the size of those polymers that are on the inside. There are two kind of methods that we use in our group to use these for sensing. One is to modify those microgels, the polymers that we use, with something to bind our target. And then we can look at how the color changes with different concentrations of that target. So a lot of target will lead to a bigger size change, which leads to a bigger color change. There's also research going on looking at binding things to the surface of the device and kind of like blocking the surface and then monitoring the response. So there's different projects going on. This process is quite complicated and I didn't work on it, so we will not talk about it, but if you do have a question, I may be able to answer it for you. For the testing that I'm going to talk about, I don't want to make you listen forever, okay. I took the etalons when they were dry, so just after they were been made. Traditionally, we would then hydrate them overnight to make sure they're all nice and hydrated, and then expose them to the target and do whatever detection method that we were looking for. The way it would be set up in the lab is the etalon would be sealed up in a little plastic chip. We would pump solution through it and use what was called a reflectance probe to monitor the wavelength of light that was being reflected. I did a lot of investigating just kind of how the devices were working. So taking them from that dry to hydrated state, kind of how long it takes, how can we make it faster, because we don't want to send people sensors and be like, you need to put this in water for 24 hours before you can use it. Hopefully you're not really sick. So how can we speed this up and make it as user-friendly as possible? And then how they respond to a target change. There's two main things that we kind of wanted to consider. The first thing is when we take the device and we change the environment that it's in. So if I take it from water and put it into like a salt water solution, you need to think about the stuff has to get into the microgels, right? They're inside that cavity, so we have to consider how things can transport in and out of the etalon device. And then how things are actually moving in and out of those particles themselves as well. So there's kind of lots of processes at play. I looked at changing the top metal layer to see if we could make them respond faster so that they would be even better to use for any of those point of need sensing applications. So the response of the etalons was definitely impacted by the thickness of the metal layer. So the green is a response to the etalon being put into a salt solution from just deionized water, so water with nothing in it. It's a nice gradual change. So basically what's happening is the top layer is providing a little bit of a barrier for that salt to enter. So it's just kind of gradually changing. If we look at the orange one, you can see it kind of like changes immediately and then kind of comes back to that nice steady state. So when we have a really thin gold layer, when we put it into the salt, there's like a rush of salt into the device, and then it kind of balances out because there's less of a barrier to it initially. This led us to be like, okay, so how big is the difference between the thicknesses that we're looking at? So these are the thinner gold films and increasing thickness of the gold that's on the top layer of the device. These are all the same scale. So you can see this scale bar is a 500 nanometer scale bar. There's a lot of cracking in this one. This film is pretty continuous, so it's a pretty solid film. So that kind of backed up what we were thinking. Okay, so that thicker film is providing much more of a barrier than the thinner films. I also looked at how this can change using different salt solutions. So you can, lots of times we think of salt, we think of sodium chloride, that's the salt that we have at home. But when we're talking about chemistry, when we say salt, that can mean a lot of different things. The key thing that I'll point out here is just using different ions in different ranging of complexity, their general size of them. But when we see a thin layer, so that's that layer with a lot of cracks, versus a thick layer, we're looking at basically the rate of response. So how fast does that color change over time? These are to the same scale. So we can see in the first 10 seconds, the ones with the really thin layer are, no matter what the ion is, are changing very quickly compared to the thick ones. So we went into a lot of stuff about this, but that's kind of the main takeaway. So these etalons are much faster with the thin ones, and it holds true for different solutions, not just one. These videos, we'll see how good the color looks. So it's just showing you kind of how fast they do change. They are running. So this one, you can see like a drastic color change kind of as that solution is passing over them. This one started before this one, and you can still kind of barely see color change happening. So when we're talking about wanting to be able to detect things at home, being fast like that is really beneficial. I also looked at using them to monitor humidity. So it's a big thing, especially for like greenhouses or food packaging industry. So when you get spinach at the store and it's gross when you get it home because it's been all wet on the inside. So there's a variety of applications that humidity sensors can be used for, as well as different vapors. There's a lot of stuff going on in research about this, especially using these devices that change color for humidity monitoring, because if we're looking at something like food packaging, if there was something in the packaging when it's like, okay, if this is blue, that means this package has really high moisture, don't buy it, it's been on the shelf forever, versus if this little square is pink, buy this one, save your money. So I wanted to see if we could use these devices that have the really thin layer to respond to humidity changes. The first quick test run we did was just to look at like human breath. So if we blew on the sensor, would it respond to just changes in our breath? I used like one of those really big straws and blew on the etalons. This one you'll notice changes quite drastically. This one I'm like blowing up my max lung capacity and can't get any change to happen. It will eventually start to change. You can kind of see on the edges first. But that led us to believe we could, yeah, so it is like a much slower gradual change on the thicker ones. So we took them and just looked at humidity in an enclosed space. So if we monitored the humidity level that was in this little chamber, could we monitor the change by monitoring the wavelength of light, but also could we see it using like a picture if we took a picture with phone, because that's ideally where we'd like to get them to give them to consumers. We're not going to send you big fancy probes and all that kind of stuff and be like, okay, look at the wavelength and find the peak and figure out what the color is. We want you to just be able to take a picture. When we track the wavelengths, this is kind of what they look like. So we have different wavelengths of light and then the reflectance. So we're looking for this peak. With the thin layer, we are able to see changes. So it goes from red to green. Can everyone see? So red here, and then as the peak goes to the green at the higher humidity, we can look at how that changes over the different humidities and if we're able to actually see changes in them. The thinner layers were more sensitive and allowed us to detect the changes better. We want to see if it was reproducible. So when we're looking at sensor research, there's a lot of different things we're looking at. Can it be used over and over again? Or is it a one and done situation? Because if it's a one and done situation, people don't want to be paying very much for it. And that leads to a lot of waste that we are already battling in our worlds today. But we want to make sure that when you use it over and over again, you're still getting the same quality of results. So we always look for reproducibility. So this is showing run 1 and 5, but over 5 different cycles of a high humidity and low humidity, the levels stay fairly constant. And we also looked at image analysis. So this would be at a low humidity, and as the humidity increases, you do get a visual color change. This is a very kind of rudimentary analysis of color. There's a couple people in the research group that do pixel by pixel analysis, and there's a lot of data and a lot of stats that I did not do. But basically, if we look at the red values, the green values, or the blue values, so that's kind of how color is described, we can look for changes. So we can see like a big drop in the blue value when the humidity reaches that 85% threshold, change in red at that 80%. It's still a very low sensitivity for the humidity, so there's lots of kind of research going on looking at if we could apply different coatings to the device that would allow it to be more sensitive to those humidity changes. But it did allow us to see that we could respond to vapors and not just use these devices in solution. And also led me to believe that we'd be able to detect organic vapors. So we're talking like acetone, so if you ever open like a bottle of nail polish remover and you can smell it a mile away, stuff like that. There's a lot of, you mentioned like A2S monitoring, right? So there's a lot of research into monitoring the presence of vapors in the air. So we want to see if we could do that with these devices. So we looked at 5 different organic vapors and just monitored to see if we could see a visual color change and get reproducible changes in their spectra. So we could, these are pretty small, but you can see that they do all change color visually. And well, except for the hexane, I can talk about that. But the rest of them gave nice color changes. Almost done. So we can see that they were sensitive to different vapors. This is actually the structure of the polymers, so the microgels that we're talking about. So there's a lot of different groups in the structure that can do things like hydrogen bonding, which is very common in these types of reactions. We can look at how polar different groups are. So polarity is something that is also very prominent in chemical reactions, and looking at how these different interactions can be happening in the polymer, because then we can look at modifying the structure of the polymers to tailor them to be more sensitive to different vapors or different chemicals. Again, with kind of being a sensor, we want it to be reproducible. So if we're looking at just turning ethanol vapor on and off, how reproducible is the color change that we're seeing? The kinetics of it. So with the system that I was using, when the ethanol was turned on, it would gradually lead to a color change. The second the ethanol was turned off, it would reset. So if you wanted to reuse the device, you wouldn't have to wait like an hour or something like that for the device to reset. And another thing that is often looked at with sensors is called hysteresis. So basically what this means is lots of the time when we do research, we start at a low concentration and then we gradually increase the concentration and we measure it as it goes up and goes up and goes up. But when you're using it out in the world, you're not going to be like, okay, I'm going to start with my lowest sample and then move up and up and up. So we want to make sure that we're getting kind of the same results, whether we're increasing the concentration or decreasing. So the gray dots are as we're increasing concentration, pink is as we're decreasing. And what we're looking for is to make sure there isn't what's called a big hysteresis. So if the gray dots went up like this, and then the pink ones kind of like went like this, that would not be very good, right? So this was pretty good. And then very quickly, this is only like a one slide situation, but I feel like I've been talking forever. The last kind of area that I wanted to use these etalon devices is for something called a strand displacement assay. They're pretty complicated, but they involve DNA. And the reason that my research group was wanting to use the etalons for this is it's typically done with an electrochemical readout, not as much with a colorimetric readout. So the system can be done basically in one tube, depending on how you do it. And it would be really easy to make it respond to lots of different targets. So I wonder if I included them. I did. Okay, so this is a better picture to show you. The way that our group was trying to do this was to take a little magnetic bead. You can buy them already made. Put one piece of DNA bound to that bead, and then we could add our target. The way the target would work is it would bind to these green pieces and kick off the red pieces of DNA. Then using a magnet, you can separate out the bead, and you would just be left with those red pieces of DNA that got kicked off your particle or your bead. And then if we had an etalon that could detect the concentration of this DNA in solution, you would basically just use the magnetic bead, hold all the stuff in the bottom of the tube, pour the liquid off onto the etalon, and the color change would tell you kind of how much DNA was kicked off that magnetic bead, which would in turn kind of tell you how much target kicked that DNA off, if that kind of makes sense. It is kind of complicated. My kind of contribution was just seeing if I could make an etalon that would respond to the DNA. Other people were working on the magnetic beads and kind of getting that going.
We've got a question here.
Yes.
What was the target in this scenario? Was it also like DNA or?
So in this scenario, it could be, okay, do you know what an aptamer is?
Yeah.
Okay, so an aptamer, does everyone kind of know what an antibody does? Yeah, so an aptamer is basically like a piece of DNA that acts like an antibody, and it can bind to different targets selectively, and DNA is just a little bit more stable usually than an antibody. So this could be something like an aptamer, so then it could bind a protein if you're looking at it. My research group was looking at THC, as well as estradiol. Estradiol is a hormone, I'm sure most of you maybe know, present in the human reproductive system. Also is a big problem in our water systems due to synthetic estradiol. Also a big interest in the cattle industry. Sometimes research goes kind of where the funding would like, as we were looking at estradiol for the cattle industry for a little bit. But the idea with this process is that I just need to change this green strand to be able to detect different targets. Instead of having to develop a brand new etalon each time, we would just have the etalon that responds to single-stranded DNA, and then we wouldn't have to make brand new etalons for all of them, or you wouldn't have to buy numerous different types of etalons. There is research that came out of our group. He basically developed this sensor using glucose test strips. So from glucose, like at-home glucose monitors, you buy the little test strips that go in. And the way those work is they have an enzyme that detects phosphate. So he was able to kind of use those test strips to detect numerous different targets, kind of harnessing this system. But the test strips are pretty expensive, and you need a certain potentiostat to measure the resistance that kind of comes out of them, because the at-home glucose monitors aren't sensitive enough. So you could use the test strips, but you couldn't use the monitor. So he was like, Haley, if you can give me an etalon that will work for this, then I can take my system that I'm kind of already working on and make it a lot cheaper. So that was the idea. My part in this was just a small project at the end, basically seeing if the etalons could respond to DNA in water. And it did work pretty well. We were able to get them sensitive in the range that he was kind of looking at with his system. So there's definitely room for improvement on this, but it is just a really cool thing that we were able to do and has lots of potential to be used for those point of need sensors. Last slide, we made it. Key takeaways. Hopefully that if you didn't know what a point of need sensor was before you got here, you kind of have an idea now, and you have an idea of where they can be applied and where they are in our world, and you've probably used one. Also that polymers are not just plastics. We have polymers all over. How simple structures like an etalons, just that simple sandwich structure, can interact with light and show promise as point of need sensors. So lots of structures like the one that I talked about today are kind of a lot of the current research in this area. And that these microgel-based etalons have kind of potential to be applied to different areas where point of need sensors are used. So with the future energy systems, I was looking at water, but also in other areas of my research looking at healthcare and things like that. That's all I have. Thank you. This is Edmonton Library, Future Energy Systems, different funding sources that kind of funded this research that I talked about today. But if you have any questions, we have time for them, I believe. So there's
We have time, but let's give Haley a round of applause. All right, the floor is yours. What do you want to know? We had some great questions during the discussion, but I will get us started.
Okay, I'll do my best.
You got this. From an environmental perspective, where do you see the biggest space that this could go in the next 10, 20 years for folks to use?
I think water is our biggest target, or was the biggest interest for us. Because water is colorless for the most part, it provides the least amount of concerns and kind of tricky problems with colorimetric sensors. So when we're looking at like something like blood or soil or those kinds of things, there's a lot of pre-treatment steps that may have to happen, whereas water, we can like directly put the sensor in it and get color change. Looking at things like monitoring people's tap water in a rural area. So we were deploying sensors into just people's homes in Athabasca that like signed up for this trial and could just monitor their tap water every like minute or so. Just like a drip would go over the sensor and a picture would go into the cloud. And if the color changed, we'd be like, hold on, stop drinking your water. We maybe didn't know what was wrong with it necessarily, but we're able to at least tell you that there was something wrong with it. But we were also looking at like even rural water, so kind of developing where it could just like float around on the lake and sample the water. So there's lots of problems that come up with colorimetric sensors, but there's also lots of areas that they can definitely be.
And can each sensor only look at one thing, or can you put that sensor in the water and it's telling you your sodium, your pH, your something else? Can it tell you all those things, or can it only tell you one thing at a time?
So currently one, but the goal is to kind of make an array of them. So I did a little bit of this as an undergrad. So we would take like a 1 inch by 1 inch glass slides. So the ones that I talked about here were 1 centimeter by 1 centimeter. So they're little squares. We can make them very small. So some of the people in my group would do 2 millimeters by 2 millimeters. So they're very small. And then you can put a bunch of them on one like chip. If you change the polymer or the antibody or whatever the target moiety was, you could then have like say 8 different ones on the chip. And it's like, hey, if square 1, square 3, and square 4 turn pink, you have this. Like that would be the goal. So that's where there's kind of research going on with different people in the group working on different targets, and eventually hopefully you can pull it all together to make them the most useful.
Awesome. All right. Any additional questions before we free you back into the winter?
Yes.
No. There are no silly questions.
Those water beads?
Yes.
Can I leave them on my plants when I go on holiday?
So you can buy them. The ones I have, I got on Amazon. Follow the instructions when it says like 1 tablespoon, only use 1 tablespoon because they expand.
There were so many of them.
They expand a lot. But the idea is, yeah, so like if your plants were like absorbing the water out of the beads, ideally. If you're planning on leaving your plants, I would suggest you can get like fill an old wine bottle and stick it upside down in the soil, and then it will
Okay.
like slowly leach out.
Okay, thank you. That's the most complicated.
I also have a lot of houseplants, so
Thank
one
you.
of the hobbies.
What's the sensitivity of this sensor? For example, if you have a water that 20%, for example, sodium, and another water is a 21% of the sodium, is it sensible that it's some changing in the color? And because this is based on eye witnesses, and you don't have any kind of image processing or something.
Yeah, so Eugene, didn't talk about any of the image processing. My good friend that I did my program with did all image processing, so like pixel by pixel, figuring out like statistically which pixels were the biggest difference and then monitoring their changes. In terms of sensitivity, like I would say you can't get that 1% right now with the way that I have these presented. Oh, I have so many animations going back, it's gonna be a nightmare. I don't even know if I showed you the calibration curve for the salt, but it is definitely not for the 1%, so there's definitely room for improvement. Lots of the sensitivity issues come from the reproducibility of the fabrication of the devices. So, because it's that metal layer, it's the microgels. You want a monolayer, so that means you don't want microgels stacked on top of each other. You want a nice, like this picture, one layer. Yeah, easier said than done sometimes. It is pretty good, but you always have a little bit going on. We're looking at different I'm not anymore, but they are looking at techniques for more consistent spreading. So right now, the way we used to make them was each person would make each individual device one at a time. That leads to a lot of human error. That leads to a lot of device-to-device error, sensor-to-sensor error, person-to-person if I'm not doing it the exact same way that you do it. And the viscosity, so the concentration of the solution of particles that you use is going to change how nicely they dry. Right now, they're working on kind of making them in a big sheet and then cutting them up so that it's more consistent. So that should help the sensitivity as well, but I don't think I had it here anyways, what I was gonna, yeah. I didn't show the different concentrations, but.
All right.
Yeah.
Is it just the top layer thickness that has an impact, or like can the bottom layer kind of?
So I did originally look at both, but if we look at, this picture's fine. This is on a piece of glass. So there isn't really any interaction with the solution through that layer. Yeah.
You mentioned the ability to reuse the etalons. I'm just wondering if like the efficiency of it will be affected in the longer run, or like the lifespan will be shortened or whatever, if it makes sense.
So the tests that I showed, I only did like 5 cycles of reproducibility. Other groups, like the ones that are employing the etalons for like the tap water monitoring and stuff, have tested like hundreds of exposures. So as long as, it'll depend on the system if you are trying to like specifically bind anything into the microgels using like antibodies and stuff like that, the reproducibility will change because you have to basically be able to undo that interaction. So something like salt response and temperature are pretty easy to change. So as long as we take it from the salt solution, put it back in water, the salt is going to leave to reach that equilibrium. But if we're looking at proteins and stuff like that, it's a lot different. So then the efficiency and things like that will be definitely different.
And the last thing that I want to say about, we should acknowledge the German scientists. First of all, the Albert Einstein, because the photonic, the first one.
Photonics.
Because the Nobel Prize of the Einstein was for photonic in 1920, and the second one is the polymer in Ziegler-Natta. The idea of polymerization from the Ziegler-Natta in 1960 after Second World War.
Yeah.
And the third one is the German engineer in Goodyear and Michelin. The cross-linking of the polymers that was from the German engineer. All these topics are the German-based.
There's very good polymer institutes based out of Germany as well, still to this day. Lots of the good core polymer research is still coming out of there.
Just think about that. your work is basically originated from the mind of the German people. Yeah.
Yep. Yeah, and polymers are so many different industries using them these days. So many different things. So.
And then the best one that the Goodyear and the Michelin, that was the revolution of the world. The Ziegler-Natta and the Ziegler and Natta was both of the Carl Ziegler and I forgot the name of person Natta. We should acknowledge them.
All right. Any last questions, thoughts, things you want to know before we leave off?
You can always ask me if you don't want to ask now, I guess.
Yes. Well, I would like to thank Haley again.
Thank you.
So a big round of applause. Sorry, I have a mic. Haley will be here to answer any questions you want to come up and ask one-on-one. Just to let you know, our February talk unfortunately had to be canceled. I know, I'm very disappointed. But our March talk will be on March 19th. It is on nuclear power, global trends, Canada's role, and future opportunities in clean energy.